Hello viewers, welcome to Mr.Wanyoike, the digital learning platform. In front of you is Mr. Wanyoike himself, a biology teacher, and from the previous lesson, we were in lesson number 2. The lesson number 2 I covered during the previous lesson, and I explained the characteristic of living organism. And today I'm going to have a review of what we had during the previous lesson. And the last characteristic of living organism covered was respiration, and I said respiration refers to the process by which living things break down the organic food substances in the living tissue to produce energy. And therefore this process is very important as far as the survival of living things is concerned. And therefore I may ask you a question. Do you normally eat to live, or do you live to eat? Of course, the answer to this question, we normally eat to live. Why do I say that? I say we normally eat to live because by eating, we normally provide the respiratory substrate into our body, and the respiratory substrate provided into our body, it will go to the tissue where it is chemically broken down to release energy, and this energy is very important. The energy produced is very important. Uses of that energy, produced during respiration. The first use of that energy is BMR, basal metabolic rate, and to control the BMR. And the second one will help in walking. The other one, writing. Whenever you are writing something, and so on, or whenever, maybe a mason, whenever you are engaged in physical activities, so you can write physical activities, physical activities. The energy is required so that it can bring about the contraction and relaxation of the muscles. And therefore, as far as that energy is concerned, it is very important for you to know that the energy generated during respiration process, it will help us maintain the basal metabolic rate. That is the basal BMR, or the basal metabolic rate. This is the energy which is required to control the involuntary activities such as blood circulation, heartbeat, breathing, and so on. The second area where energy that gotten from respiration is used is whenever we are walking, whenever we are writing, maybe whenever I'm writing something on a book, on a whiteboard, I'm using energy. And therefore this energy is very important, and therefore we cannot be able to survive without energy, and therefore respiration becomes a very basic characteristic as far as the survival of living organism is concerned. Having said that, I'm going to come to today's topic, and in today's subtopic, I'm going to look on specimen. And in specimen, as far as a specimen is concerned, specimen refers to anything that is studied in biology practically. For example, specimen, we can have a specimen in form of a leaf. We can have types of specimen. I can have a whole organism, like a whole animal. Animal can become a specimen. I can have a plant. I can have something like part of a plant, like a leaf. I can have a specimen like an orange. That is a fruit. I can talk of fruits. I can have roots can be a specimen. And as far as the other type of specimen is concerned, we can have parts of animals. can be taken a specimen like the gill, the structure of the gill from the fish. The gill, we can have the skin as a specimen, and also we can have a leg extracted from an insect. Leg can be used as a specimen, and therefore these are the various specimens that we normally use during biology practical lessons. On to having said that, it is very important to know that whenever you are using this specimen, it is very important to know that there are various ways through which we should correct this specimen from the environment. And therefore, method of correction of specimen. I'm going to illustrate methods of collection of specimen. How do we correct specimen? The method number one that I'm going to explain, we can use an apparatus. That method used during correction of specimen, and we can talk of methods and apparatus. I can include the apparatus used. Number one, we can use sweep net. Number two, the type of apparatus that we can use, we have a porter. Then number three, we can use fish net. Number four, we can have a bait trap. Number five, we can have a pitfall trap. And number six, we can have a pair of forceps. I can have a pair of forceps as an apparatus for forceps. Then number seven, I can have a specimen bottle. And number eight, I can have a magnifying lens. I can have a magnifying lens. And when it comes to a sweep net, the first type of apparatus that we normally use in biology, as far as a sweep net is concerned, a sweep net is used to collect flying insect. That is the function of a sweep net. And the second type of apparatus, it is a putter, and a putter is used to collect a small insect from the cracks of rocks and also from the bark of trees. And therefore we normally use a putter to collect small insect from the bark of trees and also from the crevices or cracks of rock surfaces. Then we have the fish net. A fish net is the third type of apparatus that we normally use during correction of fish. Whenever we want to collect fish from the water, it can be from a from a fish pond or from a liver, we can use a fish net to collect them. And also fish net can be used to collect or catch the small aquatic animals. And the second type, we have the bait trap. The bait trap is number 4, and as far as the bait trap is concerned, it is used to correct the small animals such as rodent, for example rats, and therefore we can correct rats using a bait trap. Then we have pitfall trap, that is another example, pitfall trap. And as far as a pitfall trap is concerned, this is a method which is used to correct the crawling animals, small animals like earthworm. The earthworm normally crawl on the ground, and therefore you make a pit on the ground, and they will eventually fall in the pit where you dig the pit. Then you put specimen bottle, and whenever those earthworms are crawling, they will fall into the container which is placed in the pit, and therefore after that you will have collected them. Then the other one is a pair of forceps, and as far as a pair of forceps is concerned, a pair of forceps is concerned, a pair of forceps is used especially when you are collecting dangerous small animals like stinging insect, and also when handling dangerous or stinging dangerous plants like the stinging nettle. Then specimen bottle, the work of the specimen bottle, it is where the specimen are placed after correction, and therefore specimen bottle, the size of the specimen bottle depend with the size of the specimen understanding, and therefore if the specimen understanding is of a small size, it means that the size of the specimen bottle should be small in size. Having said that, I'm going to look on to the eighth apparatus which is used during correction of specimen, and the eighth apparatus which is used during correction of specimen is the magnifying lens. And what is the function of magnifying lens? A magnifying lens contains a lens and a frame and a handle, and the lens is used for magnifying or increasing the size of the specimen. Understand it, whenever you are drawing, maybe you want to make a drawing of the specimen, you view it using a magnifying lens so that you can see even the finer details of that specimen, and therefore it helps in increasing the size of the specimen so that you can see the finer details which you could not have seen when using naked eyes. And therefore I'm going to go to the next area of study, and I'm going to caution you that there are certain precautions that you are supposed to consider whenever you are collecting specimens. And the precautions that you consider whenever you are collecting a specimen, precautions. Precaution number one, you are supposed to correct the exact number of specimen you need. Correct only the correct the exact or the correct number. Correct only the correct number of specimen you need. So there is no need of correcting excess, because you will end up leading to wastage, and therefore there will be wastage, and therefore some of them even might die before you return them into their habitat, and therefore it is very, very advisable for you as a biologist who want to study this specimen to correct just the exact number that you need during the study. And the second one, the second point that or precaution that you are supposed to consider is you are not supposed, do not harm the specimen during capture. Don't harm or injure the specimen during collection. Whenever you are collecting them, you are not supposed to injure them. And the third point that you are supposed to consider, you are supposed to handle dangerous or injurious. Handle dangerous, that is, you are supposed to handle dangerous or injurious specimen with care. So, for example, stinging insect, you are supposed to collect them using a pair of forceps, because if you joke around with them, they might sting you, and some of them are very poisonous, and therefore you will end up having an ill health after you are being stung by them. And therefore you should be very cautious, you should be very keen, you should be very careful whenever you are dealing with very dangerous animals such as a scorpion, the centipede. Those are the very, very injurious or dangerous animal, and you're supposed to handle them using a pair of forceps. And lastly, but not the least, you're not supposed to destroy. Do not destroy the habitats of the specimen. As I said earlier, I said that whenever you are collecting specimen, the specimen can be in form of the whole organism or part of the organism. And therefore, if you are collecting the whole organism, for example, let us assume that you are collecting centipede or fish, and therefore, if you destroy their natural habitat, it means that after you study, you will not get a place where you will return them, because you have already destroyed their habitat. And therefore, for continuation of their existence, it is very good for you as a biologist or as a person who collects this specimen not to destroy their natural habitat. Having said that, I'm going to come to the differences between plants and animals. We are going to look on differences between plants and animals. You can be asked, highlight the differences between plants and animals. One side, let us tabulate the differences, and here I write animals. So you have plants on one side, and the other one we have animals. And therefore, as far as this part of the question is concerned, part one, very, very important. You're supposed to note one difference is that some plants, you write, some plants have chlorophyll, that is the green coloring pigment, and in animal, number one, lack pigment, that is chlorophyll, lack chlorophyll. And this point is very, very important because some plants have chlorophyll, hence make their food. So you can write hence make their own food. Hence, the ones which lack, that is animal, hence feed on already made food. So then the second difference as far as plants and animals are concerned, we can say response to changes in the environment, or we can say that plants respond slowly to changes in the environment. Plants respond slowly to changes in their environment. And animal, number two, animal respond quickly to changes in their environment. That way. Then number three, that difference is that plant cell have cell with cellulose cell wall. And number three, as far as animals are concerned, their cells lack cellulose cell wall. Cell wall. That is the fourth one, and that's the third one. The fourth one, we can talk of do not move about. That is plants do not move about. In other words, they don't locomote. And point number 4, animal locomote in search of water and food, including the breeding mates. They can also locomote in search of breeding mates. The other difference, as far as the differences between plants and animals are concerned, we can say the difference number 5. As far as plants are concerned, we can say that plants lack specialized excretory organs. Plants lack specialized excretory organs. While animal, we can say number 5, animal have complex excretory organs. So it has complex excretory organs. And as far as point number 6 is concerned, in plants, number 6, in plants, growth occurs at the apical meristem, that is at the shoot tip and root tip, where we have the apical meristem. And number 6, as far as the growth is concerned, growth in animals occurs in all parts of the body. It occurs in all parts of the body, and therefore those are the 6 differences that we normally have as far as plants and animals are concerned. The first one is some plants have chlorophyll and therefore can make their own food. Those are through the process of photosynthesis. Those are the green plants. They normally use photosynthesis to make their own food, while animal do not make their own food, and therefore they must rely on already manufactured food. The second one is that respond slowly to changes in their environment, and in animal respond quickly to changes in their environment. Number 3, the difference is have cells with cellulose cell wall, that is plant, and animals their cells lack cellulose cell wall, and therefore the plant cell will have a definite shape because of cellulose cell wall, while in animal they lack definite shape because they lack cellulose cell wall, which prevent further expansion and also preventing the bursting of the cell. And therefore in animal cell they don't have that cellulose cell wall. And number 4, do not move about. And in other words, we can say that plants do not locomote. However, they have only growth movement, while in animals they can locomote, they can shift in position. The whole body of animal can shift in position from one position to another in search of food, water, and also breeding mates. Number 5, the plants lack specialized excretory organs, and in animal they have complex excretory organs, like it has the lungs, it has the kidney, it has the liver. Those are complex excretory organs. And number 6, growth occurs at the shoot tip that and root tip in plants, and in animal the growth normally occur in in all parts of the body, but at different rates. Having discussed all that, those were the characteristic, those were the differences between plant and animal. I'm going to leave you with this assignment. The first assignment is that you are supposed to discuss the various ways through which respiration is important as far as the survival of living organism is concerned. The various reasons why respiration is important as far as the survival of living organism is concerned. And the second question, you are supposed to discuss the differences between plants and animals. Having said that, I've come to the conclusion of today's lesson, and your presenter was Mr. Wanyoike, and you can reach me using the number as it is well illustrated here, using the number positive 254 724 901 639. That is the number which you can reach me with. That is the number that is that is positive 254. That is the code of Kenya and in my of my country. Then followed by 724 901 639. Moreover, you can reach me on the YouTube platform. Using you search me using the name Mr. Wanyoike, and also you can reach me through Facebook platform by searching me using the name MR.Wanyoike on Facebook platform. Till next lesson, bye bye.